Good afternoon and welcome to this rather extraordinary lectures on bacterial infection, antibiotics and alternatives. It's the CH2066 Major Therapeutic Areas course. There will be roughly 6 lectures, which I think will be divided into around 5 or 6 YouTube videos. We'll see how this goes. The course has 8 principal parts. Part 1, which we'll talk about in this lecture, will be the introduction to bacteria and infection and how we control bacterial growth. We'll then really focus on small molecule antibiotics and their biomolecular mechanism of action. We'll then talk about how antibiotics develop resistance. We'll look at some specific case studies. Then we'll go on to look at the class of non-cell wall acting antibiotics. Finally, we'll end up with going beyond traditional antibiotics, looking at potential alternatives. A starting point, which is particularly relevant during the current COVID-19 crisis, is to consider just how important antibiotics are to our world. It should of course be said that antibiotics have no use against viruses, so don't take antibiotics to treat a suspect COVID-19 infection. However, antibiotics do allow us to have transplant and routine surgery. Many surgeries are just simply too risky without antibiotics. For example, hip surgery and transplants, because patients have to be immunosuppressed during transplant surgery, which makes them more susceptible to infection. Much cancer chemotherapy also weakens the immune system, and patients need to be given antibiotics in order to help them survive any potential infection. And we could end up, if we don't have antibiotics, or we don't research new alternatives or new antibiotics in the future, with a situation where millions could die from infectious epidemic disease. So part 1 is introduction to bacteria and infection. So this part of course is about understanding how antibiotics fight bacterial infection and also how they stop working. We're going to look at 3 principal areas historically of the fight against infectious disease, which really have on 3 fronts: public health, vaccines and antimicrobials and antibiotics. So before we go too much further, we do absolutely need to understand the target bacteria. It's no good waging a war against the enemy if you don't understand what that enemy is. This will require some background microbiology. I do not assume anybody here has a background in microbiology or biology, but it's important that you do learn information as you go. The course is about primarily bacterial infectious disease, so I'm going to talk really about bacterial infection. So first of all, what we need to start with the basics. What do antibiotics do? What they do is they fight bacterial infection by one of 2 ways, or in some cases actually both ways, depending on the dose. They can prevent bacterial growth, or they can directly kill bacteria. You might say, why does preventing bacterial growth have any clinical effect? The reason is that most bacteria divide and grow very, very quickly. Staphylococcus aureus divides every 20 minutes, and so preventing growth can have a very useful clinical effect and give the immune system time to actually kick in and clear the remaining infection. Of course, as I said, we don't work on viruses. Before we start, I think it's useful to think what can make a bacteria pathogenic. What makes a bacteria toxic to us, humans or animals? There's a number of different mechanisms. One is to understand that they can produce toxins which directly damage healthy tissue. They can get into the bloodstream. You generally do not want bacteria in your bloodstream. Blood should be pretty much sterile, and if they do, they can overstimulate the immune system, which can lead to sepsis, or indeed understimulate the immune system, which can also lead to sepsis. And you've all heard quite a lot about sepsis in recent months. Or they can invade immune clearance and grow uncontrollably in parts of the body where you really don't want them either, particularly the lungs and the brain, etc. They may be less directly toxic in these situations, but they can still cause a huge amount of harm. This micrograph you see here is a image, I think, of Staphylococcus aureus. So what happens if you get a bacterial infection? What are both biochemical and the systemic consequences of that infection? Um, we have a primary response, which is bacteria can secrete cytotoxic cytolytic molecules, and we'll look at this in a short while. And we can also secrete enzymes, such as proteases, which break down protein, hyaluronidases, which break down hyaluronic acid in our skin, and ureases that can convert urea into ammonia. The secondary effect, the consequence of this primary infection, is the immune system response. Cytokines are released, macrophages and proteases, and a whole cascade system of immune response. And that immune response can often be as directly, or can make you feel as directly sick as a primary infection. But of course, it's necessary for clearing that, initial infection. And finally, of course, the effect of infection can lead to temperature change, which can be local, for example, at the wound site, or systemic. That's why if you get a fever, that's often as a consequence of bacterial or viral infection. And pH can change. pH can be a very useful indicator in certain situations that things are not going right. For example, urine pH should be around about 5.56. A raised urinary pH is almost always indicative of urinary tract infection. So let's start at the beginning. The first really useful mass-produced antibiotic was penicillin, and this is a poster here which says, "Thanks to penicillin, he will come home." And penicillin was the great wonder drug. It was developed, discovered in 1928, but not developed until the 1940s, really because World War II created a huge need for um, new ways to combat infection. Certainly during World War I, it's estimated that as many soldiers were being killed in the battlefield of northern France from infection following being wounded than by the initial wound or blast injury they got in the first place. But as you all know, there has been evolution of antibiotic resistance to these new drugs almost ever since the first drugs were introduced. And there's a lot of current concern about evolution of antibiotic resistance. And sometimes you will hear about the escape pathogens. These are Enterococcus, Staphylococcus, Klebsiella, Caenorhabacter, Pseudomonas, and Enterobacter. You see here some concerns even from 10 years ago, 15 years ago, about the lack of new discoveries and the lack of new antibiotics coming online. I actually am hopeful that the situation is getting a little bit better currently. Whereas in the last 5 years, there seems to be a lot more money and attention being paid to creating new antibiotics. So I think the situation is a little bit more hopeful now than it was even 10 or 15 years ago, but we will see. So let's start with microorganisms. Again, I'm assuming you don't necessarily have any biological background here, but it's an interesting place to start. And one thing to state about microorganisms is, of course, they're small. The largest are amoeba. These are things, of course, for example, amoebic dysentery. These can just about be seen under the naked eye. The next size down, which are probably tens to hundreds of micrometers across, are the yeasts. These can cause, for example, oral and genital candida, commonly known as thrush, or systemic candida infection, which is actually very unpleasant and sometimes deadly. But the next size down is bacteria. These are typically between 1 to 2 micrometers in diameter. They do vary. There are some smaller species around. And bacteria, of course, are going to be the focus of these lectures. Smaller still are the viruses, which are typically 10 to 50 nanometers across, depending on the virus. And smallest of all are the prions, which are arguably not microorganisms, but they are infectious agents. These are misfolded proteins. These are believed to be causative, the cause of diseases like Creutzfeldt-Jakob disease and bovine spongiform encephalopathy. So focusing now on bacteria, it's important to realize they come in various shapes and sizes. And the name of bacteria gives you a good clue to their basic structure. For example, spheres, known as cocci, such as staphylococci, like Staphylococcus aureus. They can be diploid, i.e. two of, such as Streptococcus pneumoniae, or in longer chains, such as Streptococci. See there, Streptococcus pyogenes. They can be rods, such as Bacillus anthracis, because of anthrax. They can be rods with flagella. You see there, Salmonella typhi, which are common food poisoning bacterium, or they can be rods which form spores. And we come on to spores towards the end of the lecture series. Spores are a very important way, but some bacteria, but not all, can evade clearance by antibiotics. And finally, bacteria can have a spiral form, such as the vibrios, such as Vibrio cholerae, which causes cholera, Helicobacter pylori, which can cause stomach ulcers. But it's important also to say and point out that we live in symbiosis with bacteria. Numerically, we have about as many bacterial cells living in us and on us as we have human cells. So we have evolved to live with bacteria. And for the most part, not only do they do us no harm, but actually they are an essential part of our survival. We would find it difficult to live in a purely sterile environment. Our gut is absolutely full of bacteria, and bacteria in our gut is used in breaking down food for the nutrients that we need. You can see here on the left some of the kinds of bacteria found in different parts of the body, everywhere from the hair right down through nostrils, the oral cavity, esophagus, stomach, into the colon, vagina, skin, etc. But in general, the primary areas where bacteria are found are across all the skin, the mouth, the colon, and there shouldn't be any large amounts of bacteria in places like the stomach, and blood should be almost sterile, as should urine in a non-diseased person. Nostrils also will be full of bacteria. And one of the main problems with bacterial infection is what is bacteria getting from somewhere where it is perhaps good or at least harmless to getting to the part of the body where it causes active harm. For example, if you perforate your bowel, the bowel is full of bacteria involved in breaking down food, but if it gets out of your bowel into your abdomen, it can cause peritonitis and in some cases rather quickly death. Clearly that's not an ideal outcome. So bacteria have can cause various adverse effects due to their enzymatic activity. They can secrete various enzymes which do different things, such as coagulase, which calculates blood. There's kinases, which digest fibrin clots involved in healing and hemostasis. Hyaluronidase, I already mentioned, hydrolyzes hyaluronic acid, a component of our skin. Collagenase hydrolyzes collagen, and also various proteases, siderophores, which are involved in our immune response, and also the way that iron is taken into cells. So bacteria can become disease-causing or pathogenic if they produce toxins which damage healthy tissues, as we said. They get in the bloodstream, causing immune damage, or they produce toxins that also can overactivate either over or underactivate the immune response. So the effects of toxic bacteria on red blood cells can be clearly seen here. This is a blood agar plate. This is a mixture of a kind of jelly mixture of nutrients for bacteria and blood. So this is, I think, probably cow's blood. And on top here, you see 3 different strains of bacteria have been streaked on top of the blood agar. Staphylococcus aureus that we have here, I'm just labeling here, Pseudomonas aeruginosa, and a non-pathogenic control strain of E. coli I'm showing here. Now, the important thing to look for here is adjacent to where the bacteria was streaked, we see this light region here, this kind of halo around the bacteria there. You see it also here around the Pseudomonas. And that comes about due to the direct hemolysis of the red blood cells by toxins secreted by the bacteria. You can really see this very clearly here. bacteria also, as well as secreting toxins, exotoxins, can also have endotoxins. And endotoxins are toxins associated with the bacterial cell wall, things like lipid A, lipopolysaccharide, and these can still be dangerous to the body, often by overstimulating the immune system when the bacteria itself is dead. When thinking about bacteria, you need to keep in mind that bacteria are fundamentally rather like us. They are living species. They communicate with one another, not of course by talking, but by small molecule messaging. They like to live in communities. Generally, they're much more successful in a community than free swimming or living by themselves. They consume resources, so they need a carbon, they need a food supply, and occasionally they have sex to pass DNA from one cell to another. Well, their primary method of reproduction, of course, as most of you know, is asexual, splitting in half. But they can, in some cases, actually pass DNA via a sex pilus. Some species can anyhow. And like us, their reason for their existence is to pass on their genes. Furthermore, they can modify their environment via enzymatic action. The point is, they are not inanimate blobs. It's very important to understand they're not simply a bag of chemicals. As I already said, most bacteria grow fast, typically with a reproduction rate of roughly every 20 minutes, or 20 to 30 minutes. There's an important exception here, however, and that are the mycobacteria, for example, which cause tuberculosis, which actually grow very slowly. This can make them very, very difficult to kill. The general way to understand bacterial growth is in terms of a lag phase, you see here, an exponential growth phase, and that's here, a stationary phase, which can last several hours, and then a death or decline phase in this final part here. People often talk about exponential phase or stationary phase of bacterial growth. Just doing some simple sums, you can realize basic genetics. You start with one cell. Within 5 generations, you have 32 cells. 10 generations, 1,000 cells. By 20 generations, you have 1,000,000 cells. So you can go from 1 cell to 1,000,000 cells in just 20 generation numbers, which in principle can just be 10 hours. So a single bacteria can rapidly produce 1,000,000 cells in 10 hours. In 20 hours, then as you can do the maths yourself, it will be many orders of magnitude higher than that. That's one of the reasons why food poisoning takes place. A small amount of contamination on, for example, a kebab purchased on a night out can rapidly become contaminated if left, for example, in a warm kitchen. So as you see here, 100 bacteria growing for 5 hours can produce 1.7 million cells. What slows down bacterial growth at the end of the day tends to be competition for nutrients and resources. So a bit of history of bacteria and disease. And arguably the modern microbiology and bacteriology began with Louis Pasteur back in 1857. Pasteur discovered the relationship between microbes and disease, immunity, and antimicrobial drugs. He demonstrated that bacteria could be destroyed by heat, and you could also prevent wine going off from bacterial contamination if you warmed it up sufficiently, not enough to boil it, but enough to kill the bacteria, what we know nowadays as pasteurization, named of course Louis Pasteur. It also has to be remembered that until the mid-1850s, it was not accepted that disease came from microbes at all. Since they couldn't really be seen, people did not accept that the cause of bacterial infectious disease was things that couldn't be seen. It was claimed, for example, it was due to miasma or bad gases coming from sewage or rivers. This lack of knowledge led to many, many deaths. Before Pasteur, in fact, Agostino Nubasi showed a silkworm disease caused by a fungus. Pasteur also was interested in silkworm disease for some reason, and he actually showed it was caused by another disease was caused by a protozoa. Now, Semmelweis was a very important epidemiologist. He's an Austrian physician working in a maternity hospital, I think in Vienna. He showed very clearly that hand washing prevented so-called childbed or puerperal fever, and led to much better clinical outcomes. However, what was interesting with Semmelweis was that this information was not acted on by the clinical community, arguably for another 50 years or more. Back in the 1860s, on to the 1860s, Joseph Lister, of the Listerine antiseptic fame, produced a chemical disinfectant based on phenolic acid to use for disinfection of surgical site infections. And that was perhaps a big step forward in terms of surgery. People started to understand how to control infection in wounds. And finally, in 1876, Robert Koch, the German microbiologist, definitively proved that bacteria caused disease, specifically anthrax, in the model he looked at in rats. And from this came Koch's postulates of infectious disease, which is still used this day to prove that a specific microbe causes specific disease. So Koch's postulates were based on There are four postulates. The first is that the microorganism or pathogen must be present in all cases of a disease. So, for example, you have 10 rats which you believe have been killed by a certain bacteria. All 10 rats, you need to be able to find that microorganism in all 10 of those rats if you believe it all died of the same disease. A pathogen can be isolated from a disease host and grown in pure culture. Number 3, the pathogen from pure culture can then cause a disease when inoculated into a healthy, susceptible lab animal, such as another rat. And the pathogen then can be re-isolated in a new host and shown to be the same as the original inoculated pathogen, for example, under a microscope. And here you see the cartoon on the right. It shows this concept. You have a dead animal, you isolate microorganisms of a dead animal, you grow them on a culture dish, you look under a microscope to identify them, you inject into a healthy animal, that healthy animal dies, and when you look at which animal was killed that second animal, and you can see here under a microscope they look the same as the original bacteria. It's a simple concept, but very important. One issue, however, about Koch's postulates is that they require a pathogenic agent to be culturable, and not all bacteria can be cultured in pure culture in the lab. And this is a major problem with his approach. But nonetheless, you have to understand that it was a revolutionary way of understanding infectious disease, and it still fundamentally underpins our understanding now. So how does an organism, for example, a person or an animal, defend against infection? So host defense can really be divided into three broad areas. The first is a kind of in effect, a wall, a physical barrier to keep bacteria out. And your skin is the largest single defense against infection. Mucosal membranes, found in your nose, also help prevent bacteria get from where they're not wanted, from where they are into where they're not wanted. But also, it has to be understood that your own microbiota, that's the bacteria that live within you, often have a very important kind of housekeeping role, and they can help to actually prevent harmful bacteria grow and spread about your body. The second line of defense can be thought of at a kind of cellular level, things like phagocytes, macrophages, and other parts of the immune system. These go around and can essentially kill cytotoxic cells, can kill infectious bacteria or viruses. The inflammatory response and associated fever are also part of host defense. Inflammation fever can raise the local temperature, or indeed the systemic temperature, to a temperature where bacteria really aren't terribly happy and grow less effectively. That's why when you have an infection, you also have a fever. Antimicrobial substances secreted by the body can also be important. And there's a whole system, part of the immune system called the complement system, which is a cascade of proteins which can recognize and target bacteria and kill them. And finally, we have what's called acquired immunity, learned immunity. We have specialized lymphocytes, B and T cells, and also antibodies which are produced in response to primary infection. Okay, part 2 of this course, controlling bacterial growth. So, bacteria can be killed, in most cases rather easily. Heat is the next way of killing bacteria. Even mild heat, such as pasteurization, about 60 Celsius, will kill most, but not all bacteria quite effectively, which is why it's used for milk and in some cases wine or even orange juice. Autoclaving involves high pressure steam, typically at about 121 Celsius, at about 5 bar for perhaps 10 to 15 minutes, is an effective way of killing virtually all bacteria and viruses, but not necessarily things like prions. And incineration is, of course, an effective way of killing any microorganism from a prion all the way up to an amoeba. Filtration can be an effective way of removing bacteria. Bacteria tend to be at least 0.5 microns in diameter, so a 0.22 micron diameter filter will remove bacteria, or at least most bacteria, quite effectively. The problem is filters clog. It does have some utility. Filters are used regularly in, for example, IV line and IV drips, and also in some milk you can buy that's been pre-filtered to remove bacteria that's present naturally in milk. Low temperature doesn't kill bacteria, it does slow its growth, which is why putting food in the fridge can control the rate at which it goes off due to bacterial growth. Deep freezing will again keep most bacteria in a state of stasis, but doesn't necessarily always kill them. Lyophilization, where you dehydrate them, is also can be a useful way to preserve bacteria in some situations, but again doesn't always necessarily kill the cells. High pressure denatures most proteins, so high pressure, particularly combined with high temperature, such as one sees in autoclaving, is a good way to denature protein and generally damage cells. Desiccation prevents metabolism. Desiccation and lyophilization are broadly the same thing, but may not necessarily kill. It does in many cases, but not all cases. Osmotic pressure, particularly osmotic pressure, can cause the plasma membrane of bacteria to distort and break, and this change in osmotic pressure combined with the weakening of the cell membrane can be a useful approach for controlling bacterial growth. We'll come on to this when we look at the cell wall acting bacteria. And radiation can directly damage oxidized DNA and other key components of bacteria, and indeed gamma radiation and even beta radiation, electron beam radiation, is often used as a way of sterilizing medical wound dressings, for example. Disinfection is a method of microbial control you'll all be familiar with. Chemical disinfectants are generally small molecule systems, and they work principally, in most cases, by oxidation or sometimes heavy metal damage to the target cell. The effectiveness of disinfection depends on the concentration of disinfectant, for example, bleach, pH, time, whatever organic matter is present. Most disinfectants are too toxic to be used directly in humans, where the can occasionally be used on the skin. If you want to evaluate a disinfectant, or indeed any antimicrobial, you can do a very simple test. You see right here, you take a porous disc, for example filter paper, you soak it in your disinfectant of choice. Here you see some examples. You see chlorine down here, phenol, hexachlorophene, and quat, which I think is also a weed killer. And here, important is, this is a bacterial lawn, marked in L, and marked here in Z, is called the zone of inhibition. And the radius of this zone of inhibition, marked here, gives you an indication, a semi-quantitative indication of the efficacy of that disinfectant. You see chlorine is effective here at killing Staphylococcus aureus. Arguably chlorine, or whatever concentration was used here, was more effective, for example, than quat. If you look at this E. coli bacteria here in the middle what we can immediately see is chlorine again is effective at controlling bacteria by the zone of inhibition, but quat and hexachlorophene are relatively ineffective. In fact, hexachlorophene, by the absence of this halo, the zone of inhibition shows it basically does not kill E. coli, or a particular strain of E. coli. And even more strongly, you can see with Pseudomonas aeruginosa, gram-negative bacteria, chlorine is somewhat effective, less effective than it is on Staphylococcus aureus, but the other disinfectants here are entirely ineffective. And this also points to a general thing, a general observation, but gram-negative bacteria are harder to kill than gram-positive in most, but not all cases. The different types of disinfectants, the ones which can be used typically on skin, and you might be contained in, for example, contact lens solution, things like the biconides as chlorhexidine. These are these long molecules that disrupt the outer membrane of a bacteria. Then next we have halogens, iodine, chlorine, etc., which are basically oxidizing. Bleach, for stuff you might put in your toilet, is hypochlorous acid, for example, and will directly oxidize target organic matter. And then we have the alcohols, such as ethanol, isopropanol. These work by essentially just dissolving away the cell membrane on the outside of the bacteria. Dissolve the lipid and the bacteria cell falls apart. Finally, heavy metals, such as silver, mercury, and copper, work by denaturing proteins, possibly binding to specific, in case of silver, cysteine residues which contain sulfur within the cell itself, disrupting its function, possibly rendering bacteria less amenable to coping with normal oxidative stress. It can also disrupt the plasma membrane, and as I said already, here you can see disrupt enzyme function. So silver, mercury, and copper. Silver is now commonly used as an antimicrobial, although the clinical evidence of efficacy is somewhat lacking. Copper is not generally regarded as particularly effective heavy metal disinfectant, and may have some utility in some situations. Mercury is effective, but of course mercury is also highly toxic. So as I already said, most disinfectants are too toxic for internal use. You wouldn't want to drink bleach, for example, it would kill you. And neat bleach is too toxic for external use. However, you can use diluted bleach in some situations. Some disinfectants may be effective antimicrobials, but in general, chlorinated phenols are best avoided. Even TCP, which you can purchase over the counter, although it's an effective disinfectant, may actually be a cancer suspect, a suspect carcinogen. My personal advice to all of you is actually not to use TCP for anything, because it's almost certainly harmful. There's strong evidence it's harmful. The reason it's still around is it has what's called grandfather rights from a regulatory perspective, i.e. it's been around so long that it's just been accepted, although if it was ever produced today as a proposed disinfectant, it would never be allowed on the market. So the fundamental challenge in designing any antimicrobial, an antibiotic, or indeed even a disinfectant, is to use something that is less toxic to eukaryotic cells, which are what we are primarily made of, than prokaryotes, which are bacteria. We'll come to different eukaryotes soon. That's particularly important, of course, if you intend to swallow or otherwise take in your antimicrobial. This poster here on the right is, I think, from the 1930s, used in the US Army. It said, "A minute with Venus is a year with Mercury." Venus being, of course, the Greek goddess of love, and a common sexually transmitted disease was syphilis. And the only cure for syphilis were mercury-based drugs, which were themselves highly toxic. However, arguably it's still better than dying of syphilis. So let's begin thinking about the war against infectious disease from a broad perspective. And I think we can really think about it in terms of in 6 broad areas. The first is public health. That's provision of clean drinking water and the removal of sewage. We then have antisepsis, which was really developed from the mid-19th century onwards. Things like carbolic soap, phenolic acid, that kind of thing. We already talked about Koch and Lister. Also, of course, Florence Nightingale for understanding the need for cleaning, particularly hospitals, to prevent infection. It should also be said, of course, that the ancient Greeks and Romans understood the importance of clean drinking water in public health, particularly the Romans, and went to some effort to provide sources of clean drinking water in, for example, Rome via aqueducts. Vaccination is hugely important. Since the 1950s onwards, vaccination has been routine. Polio, MMR, etc. have had a massive positive benefit on the war against infectious disease. Antibiotics, really from World War II onwards, penicillin, and then a whole other large number of other classes of antibiotics have been developed. Antivirals have had a slightly more checkered history, but they do appear to be now effective against HIV, and possibly some strains of flu appear to respond to certain antivirals, such as Tamiflu. And then we move into a new area of, for example, peptides, phage, which are viruses which kill bacteria, and possibly biological therapies based on immune stimulation may come in the next 10-20 years. The relative order of importance of this list can be debated. My personal view, from looking at from a whole society perspective, is probably that public health is the single most important, has been the single most important intervention against infectious disease, arguably followed by antisepsis and vaccination. You might, from a numerical perspective, antibiotics perhaps have been less important, but that's not to negate their importance overall. This is a graph here, which is an interesting good news graph. The left-hand axis, the y-axis I should say, is deaths per 100,000 persons, and it shows that in around in 1900 there were 400 per 100,000 people dying of some kind of infectious disease, and that has come down very, very rapidly right up to roughly 1955. So it's been a good news story. The spike you see here is due to the influenza epidemic in 1918-1919 at the end of World War I. Okay. The things here, for example, pertussis is our vaccinations which were produced. Whooping cough, pertussis 26, tetanus 27, and polio, Salk vaccine, remember Sabin polio vaccine in 55 and 62, and measles, mumps, rubella, and so on and so on and so on. So there's been there's arguably a lot to be happy about in terms of the infectious disease, although our current situation with COVID-19 may produce an unfortunate and unwelcome spike in this overall decline up here in 2020. So let's start with public health. This is really about sewage and clean water. It may seem perhaps obvious to us, but it wasn't until John Snow, the famous epidemiologist in London, showed the link between contaminated water and disease that people started to really take provision of clean water, getting rid of sewage seriously. So John Snow was really arguably the great grandfather of epidemiology. And what John Snow showed, this is in a working class part of London, in Soho, it's not working class anymore, he plotted on a map the number of deaths, and the same map was all from cholera, from the great cholera epidemic in 1854, and also the situation, the location of pumps. You can see these pumps here in black. These are water pumps. Of course, in those days, we didn't have taps in the houses. They would go along to the local pump and extract water. The problem here was the groundwater had been contaminated with fecal material from cesspits, which were many in the locality, particularly this pump here. So John Snow showed this causal relationship. He then went along and, as legend had it, if it's true or not, he actually removed the handle from the pump. You can actually see the pump if you walk through Soho today with its pump handle removed, and in doing so prevented a large number of deaths from people pumping up contaminated water from that particular well. It's a hugely important kind of real-life experiment. Okay, part 3, small molecule antibiotics. This is where we're really going to start to focus on the chemistry side of this course. The first part's really been I think, an important introduction. Now we're going to kind of zoom in and talk in more detail. So when I say small molecule antibiotics, these are really the kind of molecules which you almost all listening to this will have been prescribed at some point in your lives already for bacterial infection. Okay, I'm going to talk a bit about the process of drug discovery for antibiotics, although some of this is pretty generic and you have seen for other diseases, not just bacterial infectious disease. So let's start with the fundamental concept of drug design, which I already mentioned, but needs stating because it's easy to forget as a chemist. And that is the drug needs to be more toxic to the target than host tissue. It's stating, arguably stating the obvious, but it doesn't need stating. So how do you do this? How do you make a drug? How do you design a drug to be more toxic to a target than host? In terms of bacteria, you exploit differences between the eukaryotic cell, which is basically, as I said, us, and prokaryotic cell, which are bacteria. And specifically, you look at the fact that in bacteria, they have in general one circular chromosome, which is not membrane bound, they have no nucleus. They have peptidoglycan cell walls, and we'll look at cell wall structure soon. They reproduce primarily by binary fission, and also they don't contain cholesterol in the membrane, which could in some cases be important. Eukaryotes, which are what we're made of, well, the non-bacterial parts of us, of course, have paired chromosomes, 46 chromosomes, 23 pairs, which contain all the genetic material, almost all the genetic material, that's not all, but most of it, and it's contained within the nucleus. The ribosome, which is the protein factory in cells, is different, has a different structure in a eukaryote than a prokaryote, and this difference in structure we'll discuss in later lectures is an important thing that we can exploit in antibiotic design. And also importantly is the cell wall in a prokaryotic cell is different than in a eukaryotic cell. Again, this difference can be exploited. So antibiotic agents. The background to antibiotic therapy predates microbiology, and it was really came from the observation that some organisms can kill other ones. In other words, you co-culture, for example, two bacteria or a yeast and a bacteria, and you can see that one inhibits the growth of the other. This is called antibiosis. In fact, the Chinese realized this 2,500 years ago when they showed that moldy soybean, I think what we probably call tofu nowadays, put on boils would have a useful therapeutic effect at reducing infection. As already mentioned, Pasteur noticed that anthrax failed to multiply and died if co-cultured with certain bacteria. And indeed, the birth of the modern age of antibiotics came about due to the observation made by the observation that penicillium mold inhibited the growth of Staphylococcus aureus, and we'll come on to look at this right now. A little bit of a word on nomenclature. I've probably already been a little bit not as strict as I should have been about nomenclature, but we should be clear what we mean. Antimicrobials is an umbrella term for any substance which controls the growth or directly kills a microorganism. Strictly speaking, an antibiotic is a naturally derived substance, but it may be made synthetically, which controls the growth or kills bacteria, but not, of course, viruses. Nowadays, there are entirely synthetic antibiotics, which are not naturally derived, but are still termed antibiotics. So this definition is now already a little bit suspect. An antiseptic is a substance which, when applied to living tissue, can be applied to living tissue for purposes of controlling microbial growth. Bleach, for example, is an antiseptic. It would, although only if it's sufficiently dilute, but it doesn't cause harm at the same time. And just to remind you, an antiviral will target viral infection. So where did it all begin? Arguably in 1928, Alexander Fleming discovered that the mold Penicillium, this fungus, it's a mold, would had a zone of inhibition around it. And you see here with culture plate, there's a penicillium mold. This is a mold I'm showing here. There's a very nice clear zone of inhibition when Staphylococcus aureus, we see here, bacteria, has not grown. Legend has it that Fleming was not the tidiest of scientists, and it was a bit of mouldy bread that he had lying around in his lab, which contained the penicillium mold, and he just happened to notice that it got onto a culture plate and prevented bacterial growth. So it's a great observation. One interesting thing about Fleming was apparently he's a terrible speaker, very shy, very poor at communicating, which meant there's a large time lag from 1928 when the discovery was made to 1940s, the early 40s, when it was realized that penicillin, which is the small molecule derived from penicillium, started to be mass produced, and used for fighting infection. You might say that this a poor ability to communicate costs probably millions of lives. So that would be a little bit unfair on Fleming, but it was it still paused for thought. So next time you're in London, go by train, get off the train to Paddington. Here's Paddington Station, here you see on the left. Here's Parade Street. You come out of the station, far left corner, walk along about 100 meters perhaps, and look up. And there you see Fleming's lab. And I still think it's quite amazing that we have this great godfather of antibiotic therapy just a little over 18 minutes away from us in London on a fast train. That's the St Mary's Hospital on Parade Street. So a bit of history of antibiotics. As I already said, penicillins came about in the 1940s. In the 30s, there were a group of drugs called the sulfonamides, which were enzyme-acting antibiotics, but they weren't desperately effective. But they were still should be mentioned. Penicillin is actually, in fact, part of a family of a more general family of antibiotics called the beta-lactams, and you'll see why shortly, because they contain a beta-lactam four-membered ring. In addition, in 44 was aminoglycosides we'll discuss in a while, chloramphenicol and nitrofuran. And in this graph you see on the top are the antibiotics. And importantly, you can see how resistance started to occur. So within a few years of the first antibiotics, for example, the beta-lactams being introduced, already, in fact, even before industrial production of penicillin, what's called penicillin A's, which is an enzyme that degraded penicillin, had been identified. Sulfonamide resistance was already seen in 1942, following its introduction in 1935. Streptomycin resistance, which is one of the aminoglycosides, resistance was seen roughly 3 years after it was introduced. And this pattern of a new antibiotic coming on the market, coming into general use, and resistance being seen, is a general feature of new antibiotics. In the 50s, we had the nitromidazoles, glycopeptides, streptogramins, tetracyclines, quinolones, all of which are still in use today. Although, and again, we see here that macrolide resistance was seen in 53. The first resistance to methicillin was seen in 61, and so on and so on. Then there's a bit of a gap in new antibiotic discovery. Really, from the 60s, there's not a lot that came on the market in the 70s, 80s, 90s. But then we came into the 2000s, and we have new lipopeptides and oxazolidones, which are an important new class, probably one of the most important new synthetic classes of antibiotics discovered in mid-2000s. But already, as you can see here, we already have resistance occurred shortly after their introduction. So the effectiveness of an antimicrobial treatment really depends on a number of different variables. One is the number of bacteria causing an infection. If you have, for example, a cut in your hand and it's contaminated with a small number of bacteria, it's fairly easy to control that infection, for example, just by topical antisepsis or even just using soap and water in the wound. If that same wound is contaminated by a very large number of bacteria, it will be much harder to eradicate an infection. Also important is what the bacteria are doing in that wound or that environment, and also for example for temperature, and the fact that biofilms, which are bacterial mats, are a very important way in which bacteria evade immune clearance and antibiotic action. Time of exposure to bacteria is important. Basically, the longer, the worse the outcome. So the sooner you can get a wound, for example, clean, or an infection treated with an antibiotic, in general, you get much better outcomes. And then, of course, the bacteria themselves, microbial characteristics, have a very large effect on the overall prognosis. Also important is the efficiency of transport of the antibiotic or antibiotic to the infection site. This is why bone infections can be very difficult to treat. Bones don't tend to have a very high blood supply, and in general we rely on blood to supply our chosen chemical to the site where it's needed. So parts of the body which are not well vascularized tend to be hard to treat when they become infected. Also important, well, not stated here, but I need to state it now, is the immune response is really fundamental. If you have a healthy immune system, you will generally have a good chance of fighting off an infection, often without an antibiotic or an antimicrobial. But if you're immunocompromised, it can be much, much more difficult to fight off the infection. A little bit here on understanding and quantifying antimicrobial efficacy. What's often discussed is the so-called MIC of an antibiotic. This means the minimum inhibition concentration. That's the least amount of the molecule or your choice required to inhibit growth. Not kill, but inhibit. Here's an example of some data, some molecule we made, zinc and copper SB. I think the zinc shift base, the copper shift base, as I recall, and we measured their antimicrobial efficacy against Staphylococcus aureus, MSA476. And what you can see here is our zinc system has a much better MIC roughly 500 micromolar, apologies, compared with the copper system, which is probably 600 micromolar. What we're measuring here on the y-axis is the optical density at 600 nanometers. We're basically essentially measuring the density of bacterial cells seen by how much that planktonic cells scatter light. If you look here on the right, what you can see is the effect of our copper or zinc system at these different concentrations. You see a high concentration, and I just pointed that out here. You see it looks clear. Well, it's coloured, of course, because the broth is coloured. You can see it actually looks, although it's a clear yellow liquid. But just 100 micromolar lower concentration, so that will take us the zinc system from that concentration down to that concentration for 500 there to 400 there. You can see we get appreciable. It looks appreciably cloudy. At 300 micrometers, it's very cloudy, so that's about there. And that's the optical density that we're measuring. So this is optical density, very easy to measure. It's a UV vis measurement, but at a fixed wavelength. You just measure how much light is transmitted. It's semi-quantitative. It's not a very good way of quantifying bacterial density, but it's a useful and quick look and see for whether you are getting some overall bacterial clearance or prevention of bacterial growth. And you can also discuss MIC in terms of MIC 100%, sure you get complete inhibition of growth, or MIC 50, for example, which would be about here, where you've reduced your bacterial growth by 50%, or MIC 90, which would be about here. Also, you always need to define which MIC you're talking about. To say, before we go to the next slide, but people also talk about MBC, which is minimal bactericidal concentration, which is the amount of antibiotic required to kill the bacteria. I mean, in general, the MBCs are much higher than the MICs. So, what are the characteristics of a good antibiotic? So they should selectively kill or inhibit growth of bacteria at low concentrations, especially by low MIC. And as a rule of thumb, a good small molecule antibiotic that you would take systemically would have an MIC perhaps of 5 micromolar or less. So you can clearly see that the zinc SB and the copper SB systems I showed on the last slide are very poor antibiotics, having an MIC roughly 2 orders of magnitude higher than what we would expect for a good antibiotic. They should be in principle broad spectrum. That means they can kill many different species and many different strains of target bacteria, and preferably gram-negative and gram-positive bacteria. There is some discussion about whether broad spectrum is a good thing, because it may increase the rate of evolution of bacterial resistance. Clearly, you want minimal side effects. You want minimal side effects due to the antibiotic itself. That can be the direct cytotoxic action of the antibiotic, or its toxic effect from metabolites when it's being eliminated, particularly on the kidneys. Many antibiotics can be quite nephrotoxic, kidney toxic. You also need to be concerned about the effect of other, if you like, good bacteria on the body. You really don't want to eliminate, for example, all the good bacteria in your gut if you can avoid it. And then you also need to worry about pharmacokinetics. That basically means getting the right drug, the right place, at the right time, at the right concentration, and then getting it out again in a way that doesn't cause harm. We'll come on to pharmacokinetics soon. We also need to concern ourselves about the distribution and administration mode. Ideally, antibiotics can be taken orally, but not all antibiotics can cope with low pH conditions in the stomach, and therefore have to be injected, which is in general less desirable.